My name is Andrew Conway-Morris. I'm a Wellcome Trust Clinical Research Career Development Fellow at the University of Cambridge and a consultant in intensive care medicine at Addenbrooke's Hospital, Cambridge. I'd like to introduce you to my recent paper, C5a impairs phagosomal maturation in the neutrophil through phosphoproteomic remodeling, which was recently published in JCI Insight. The first author on this paper is Alexander Wood, a Gates scholar who recently completed his PhD with me and has now moved to Melbourne, Australia to continue his clinical and scientific career. One of the major problems that faces my patients in intensive care unit is its secondary infection. That's an infection they acquire whilst on the unit. In recent years, myself and others have demonstrated that these secondary infections arise as a result of failure of immune cell antimicrobial functions. On the face of this immune cell failure is counterintuitive, as many of the patients in ICU suffer from excessive immune activation and immune-mediated organ damage. Back in 1992, Peter Ward and colleagues in Michigan started to unravel this paradox, demonstrating that pro-inflammatory molecules, and particularly the anaphylotoxin C5a, could drive dysfunction in neutrophils in animal models of sepsis. More recently, I demonstrated this in critically ill patients and showed that this preceded and predicted subsequent secondary infection. Neutrophils, as the largest population of immune cells and the first responders to invasive infections, are key to defending against secondary infection. Complement activation is a feature of a range of infectious and sterile insults, with the cascade of plasma enzymes catalysing the formation of convertases, which ultimately lead to the generation of the membrane attack complex that punctures the cell walls of targeted microorganisms and leads to cell death. Along the cascade, various split products are released which have inflammatory functions, most notably, for the purposes of this paper, the anaphylotoxins, and particularly C5a. C5a can also be generated directly by activated innate immune cells, including macrophages and neutrophils. In sepsis and other forms of critical illness, systemic activation of complement and massive release of C5a occurs, leading to immunoparesis. However, the mechanisms which underpin C5a-mediated neutrophil dysfunction remain incompletely understood, and we lack effective treatments for this problem. This paper is built on two novel assays. The first was a model of bacteremia, which is where bacteria enter the bloodstream and which leads to severe illness. We used healthy donor blood anticoagulated with the direct thrombin inhibitor, argatroban, which does not interfere with either complement activation or immune cell function. The bacteria were two common secondary infecting pathogens, namely the gram-positive Staphylococcus aureus and the gram-negative E. coli. These were labelled with a pH-sensitive dye, which allowed them to be detected once ingested. The use of an Attune acoustic focusing cytometer allowed for unlysed whole blood to be run, so minimising manipulation artefact. In later variations of this assay, we combined the pH-insensitive Alexa fluor dye with a pH-sensitive pH-rhodo dye, allowing us to distinguish between ingestion and acidification. The second novel assay was a new system for profiling the neutrophil proteome and phosphoproteome. Neutrophils, as the shock attack troops of the immune system, are packed full of proteases and phosphatases, and if not handled correctly, will rapidly autodigest their content. However, trypsin digestion is key to the generation of peptides for proteomic analysis. Working with colleagues from the Cancer Research Cambridge Institute Proteomics Core, we developed a neutrophil-specific system that provided unprecedented depth of the proteomic and phosphoproteomic data for neutrophils, with 4,859 unique proteins and 2,102 phosphoproteins identified. Using our model of bacteremia, we demonstrated that C5a could impair the ingestion of Staph aureus and E. coli, and that the temporal sequence of C5a and bacterial exposure matters. It is only if the cell encounters C5a before the bacteria that phagocytosis is impaired. Importantly, we demonstrated that C5a induces a prolonged defect in phagocytosis. Following pulsed exposure, this persists out to 7 hours after the C5a has been removed. This implies prolonged C5a signalling and supported our previous contention that defects seen in patient neutrophils after several hours of ex vivo processing were capable of having been produced by inpatient C5a exposure. This data led us to ask what persistent signalling pathways are activated by C5a and how does this impact on the signalling following exposure to Staph aureus. When neutrophils encounter Staph aureus, there is a rapid and profound change in the phosphostatus of hundreds of proteins, 863 showing significant change in phosphostatus, and these mapped to hundreds of pathways. However, when C5a exposed neutrophils encountered Staph aureus, there was a marked reduction in the phosphoprotein response, and 19 proteins were significantly different between the C5a and control conditions exposed to Staph aureus. These mapped broadly to two pathways: vesicular trafficking and nuclear reorganisation. Of particular interest was this protein, ATP6V1G1, which is a component of the vesicular ATPase proton pump, which is responsible for acidifying the phagosome. As our initial work had used a pH-sensitive dye, this begged the question whether the effect we saw was due to a failure of acidification or a failure of ingestion. In order to answer this, we developed a dual-label probe with an Alexaflor 488 and pH-Rhodo conjugated Staph aureus. As you can see in this video, the green particles are ingested and then become progressively more yellow as the pH decreases within the phagosome. In this flow plot, one can see the cells moving along the Alexa 488 x-axis as they ingest particles, before moving up the pH-Rhodo y-axis as they acidify. Crucially, what we see following C5a treatment is the persistence of this population here in the bottom right corner, which is cells that have ingested but not acidified their phagosomes. So C5a induces defects in both the ingestion and phagosomal acidification and maturation. So how well does this in vitro exposure to C5a replicate what happens in critically ill patients? To answer this, we worked with a long-term collaborator of mine, Professor John Simpson from Newcastle University. Using the same bacteremia model as we had developed in Cambridge, we exposed blood from critically ill patients to the dual-labeled probe. Here, we can see that neutrophils from patients that had dysfunctional neutrophils, classified using our well-established and validated Zymosan ingestion assay, had impaired phagosomal acidification, whereas those from patients without dysfunctional neutrophils had similar acidification to a group of healthy donors. What is the importance of this study? We demonstrated that C5a can induce persistent and widespread alterations in the neutrophil signaling response to bacterial pathogens, and that this defect extended beyond what was already known about particle ingestion to show inhibition of phagosomal maturation. This leads us to suggest that a failure of bacterial clearance by dysfunctional neutrophils may not be simply a failure to ingest them, but also a failure of intracellular killing. This may have important implications for the role of neutrophils as vectors for disseminated infection. Thank you for watching my author's take on our study. I'd like to acknowledge the various people who contributed to this manuscript and also the funders, the Wellcome Trust, Gates Cambridge, European Society of Intensive Care Medicine and the Academy of Medical Sciences. Thank you for watching.